like to welcome you all to the next talk in our How To Talks by Postdocs series at the Health Sciences Library System, University of Pittsburgh. Today we have Dr. Caitlyn Barrett from the Hastings lab where they work on Parkinson's disease. Today Caitlyn is going to be giving us a primer on molecular cloning.
Thank you very much. So, that was a pun intended, a cloning primer. Their primers are really important for cloning, so good start. I'm trying to be funny. Okay, so what I'm going to do today is give you a very basic outline on how molecular cloning works. Then we're going to go through an example using some technology that the Health Sciences Library actually has available for you guys. And I'll be able to show you exactly how I would go through the planning process of cloning, going from having an insert that I want and having a destination vector that I want and combining the two. And then I'm going to, once you know the theory, I'm going to actually show you some tricks. So cloning can be a lot easier than it seems and than it has been in the past because there are a lot of new systems that have been developed that make cloning as easy as popping an insert out of one place and into another place. So that's what we're going to basically be doing today. The first thing that you would want to think about when starting a cloning project is, why am I cloning? So the purpose of cloning is making it so you can express your gene of interest in any way, in any manner that you'd like. So the questions that you'd ask yourself is, do I want to have a tag on my protein of interest? Do I want my protein of interest expressed alongside a GFP so that I can see which cells I've transfected and things like that? And how much do I want the expression to be occurring? Do I want to use a really strong promoter that's going to cause overexpression like gangbusters, or do I want to use a very weak promoter that's going to be like more of an endogenous overexpression level? So those are the kinds of things that you want to think about when you're deciding how you want to express your protein of interest. So basically, why we're cloning is so that we can exogenously express a protein of interest, and so that we can tag it or co-express a marker or change the promoter and also titer the extent and location of exogenous overexpression. So if you have cells that you want to see if your protein of interest is protective against oxidative stress, things like that, what you're going to want to do is be able to overexpress your protein of interest, and that's where cloning is really important. So the things that you need, very basically and overarching, are number one, a destination vector. And this is where really thinking about how you want to overexpress your protein of interest is important. So in your vector, this is an example here. This is a vector that has a GFP on it. That's the GFP. It also has a promoter that's good for expressing shRNA. All these things are things that you'll want to think about before you pick your vector. But you'll have your vector that you want to put your gene of interest into. And basically, you'll want to look for your vector based on what exactly you want to do with that vector. You'll also need an insert source, so you can get your insert source from genomic DNA or cDNA made from mRNA. You can get it from another vector that maybe doesn't have the tag you want, but it has the gene of interest that you want in it. Or for very small constructs like shRNA, you can actually have oligos made by a company like Invitrogen. So those are your options of how you get your insert source, and then you also need restriction enzymes that are specific to your project. So I'll get more into detail about restriction enzymes, what they are and why they're important. But you'll need restriction enzymes specific to your project, and then really importantly, lots of planning. The actual cloning process takes probably an hour hands-on time, a couple of days in general. The planning process should take double that time because you want to make sure that everything that you're doing has been done in silico before you ever get to the point where you're actually making your construct. So for destination vectors, this is the simplest part. All you need to do is figure out what you want and how you can get it. And a lot of destination vectors are available through several different mechanisms. You can look at nonprofit plasmid repositories. So these are places where another scientist submits their sequence or submits their plasmid, and these companies hold on to them, and they put together libraries, and you can search for them, and you can get much cheaper plasmids that way. And then there's also companies that offer plasmids, Origin, Genecopedia, and Sigma. And I actually gave you a list, and if you didn't get one, I've got one here, but a lot of different resources that you can use to find these different things. But basically what you can do is, if we say went into the Add Gene plasmid repository, you can go to Find Plasmids. You can search, you can browse. I mean, a lot of times when I go into something like this, I say, well, I want high expression and I want a GFP marker, but I'm not 100% sure how exactly I want that plasmid made up. So sometimes what I'll do is go to these different filters. I want mammalian expression, and I want it to be, let's see, I want to do one insert plasmid type encoding. The insert species doesn't really matter because we're going to be putting the insert into there. Antibiotic resistance, and you can start paring down what exactly, what they have as options. One of the things that I like to do too is, if I want a GFP marker, search for GFP and look at the different options that they have. So here is an IRES GFP, so it's going to express GFP at the same time as your protein of interest, but it's not going to have a GFP tag. So different things like that, and a lot of times your boss will, or you will already know why you are doing this. Say you want to see where is this protein expressed in certain cells, put a GFP tag on it, and you'll be able to see in real time where exactly that protein is going. So you'll know exact or kind of what you want to do, and you can start searching for plasmids that way. Okay. So that's the easy part. You find the plasmid you want, you order it, and now you've got an empty plasmid that you can pop whatever you want into. The big thing for cloning is figuring out your insert source. So, some different ways you can make those inserts. I mentioned them earlier, but the first thing you can do is cut or PCR amplify your gene of interest from a vector. So say you've got a vector in your lab, it's a pcDNA 3.1. So just a plain old overexpression vector, but you want a GFP tag. So you can take your gene of interest right out of that plasmid. And the way you can do that is either by cutting with restriction enzymes, again I'll get into that a little bit later, or you can use primers to your gene of interest and amplify by PCR what you want to have. So you can end up with these nice PCR amplified duplexes that you can ultimately pop into your vector. And I also mentioned for shRNA, you can do synthesized oligos. So basically, if you have an siRNA sequence, you can put that into, you can pretty much put it into a Word document, and then put in a loop sequence, and then put in some restriction sites, and basically you can say, in Vitrogen, can you make this oligo for me? And you make the primary strand and the reverse complement, and you can clone it from there. That's a really easy way to do that, but it's only for oligomers that are 90 nucleotides or less, so smaller things. And then you can also PCR amplify your gene of interest from genomic DNA or cDNA that's been made from mRNA. So basically what we have here are the two options. You can isolate your DNA, select primers that are specific for your gene of interest, perform PCR amplification, and again you get this nice little block of your gene of interest. It's a little easier because you avoid having these super long sequences with introns and exons to just use the mRNA. So basically you can use a reverse transcriptase, make cDNA from your mRNA, which is just your open reading frame, just your coding sequence, and ultimately PCR amplify from your cDNA and get your insert source. And we'll talk more about primer design and everything in a few minutes. So once you have your insert source, you have to think about what restriction enzymes that you're going to need that are specific for your project. So broadly, cloning is this easy. So basically what you have is you have your vector that you want to go into, you have your gene of interest, and it has these restriction sites on it. So these are just portions of the DNA that are sequences that can be cut by restriction enzymes. In this particular example, this is XO1 and EcoR1. That's not important right now, but basically what you do is you digest with these restriction enzymes. You have these nice little overhangs that are compatible with one another, and you can ligate these together and ultimately get your CD or your plasmid with your gene of interest within it. So simple. So why are we having this class anyways? It's not really that simple. Again, we get to the planning part. So what you're going to need to do, here we are at the restriction enzyme portion, what we need to do is figure out, number one, how our destination vector can be opened up so that it can accept our insert into it. So most of the time, the plasmids that we have what are called multiple cloning sites. And this is basically a huge run of a whole bunch of different restriction sites so that you can open your vector and insert your insert into there. So this here is a multiple cloning site. It's got all these different restriction sites in it. Restriction sites are these little small pieces of DNA that are recognized by restriction endonucleases. So restriction endonucleases occur in bacteria normally. And they're used to get rid of viruses that are infecting them. So virus DNA just happens to have these restriction sites in it, so whenever it attacks a bacteria can release the restriction enzymes and break down the viral DNA. We've hijacked that process, and you can input these sequences into a plasmid so that you can use these restriction enzymes to cut the plasmid open. And they all cut in different ways. We have some cuts that are called blunt, and it just cuts right in between two nucleotides all the way through the duplex. Other ones leave these sticky ends, and these are really optimal for cloning because when you cut your destination vector with these restriction enzymes, they leave that sticky end. So basically you're going to get this end just floating around. If you cut with the same enzyme, your insert, it'll also have a sticky end and they can hybridize to one another. So that's really the trick for basic cloning, is being able to use these restriction sites. So in this case, for this pcDNA3, if you said, I want to use the HinD3 and the EcoR1 restriction site, that's what you're going to design your insert based on. And what you can do that's really nice is go to New England Biolabs. They have all of the restriction enzymes. They also have this really cool tool called the Double Digest Finder. So if you decide, I want to cleave with EcoR1 alphabetically, and you want to cleave with HinD3, you can just go through here and press go, and ultimately what it'll give you is the conditions that you use for that digest. So when you're digesting your plasmid, you will do it in the particular NEB buffer at 37 degrees, and they tell you how much time, all that jazz. So that's a really easy step, and you can get all of your restriction enzymes from New England Biolabs, and they send with it the buffer and everything like that. So that's actually one of the places that's on this cloning cheat sheet I gave you, so that you can look up different, it'll also tell you what the restriction sites look like and things like that. Alrighty, cool. So now you've got your restriction enzymes that you've chosen, and ultimately we're getting to the planning phase. And this is so important. I can't even begin to tell you how important, because you don't want to go through the cloning process, the validation process, and find out that you have got, you don't have anything in your plasmid, you don't have your insert, or that there's a frame shift and you can't overexpress your protein of interest. So this is where some tools are really important for you to have. Number one, a powerful sequence analysis program is really important. So Vector NTI is the one that I'm going to be using for the examples in this presentation. Pitt has a license agreement, and you can get that for free on any computer at the university. CLC Sequence Viewer is also free, but I just don't think it's quite as powerful as Vector NTI. And then primer design software that can be found online, like Primer 3, is also really helpful for double checking your design of your primers. But this is a plug for the HSLS, and that is you can go into their website and you can search for Vector NTI Advanced, and you can download this program for free, and it's wonderful. All of these examples will be based on that. Okay, so this is our example. I am going to take you from having a vector that I want to put my gene of interest into and a gene of interest, and show you how I can plan the cloning process. Okay, so what we're going to work with here is a very simple plasmid. The pcDNA plasmids, if I were to be starting a cloning project and it was a very simple one, like adding a tag or something, I would stick with this family of plasmids because they're very simple, and most of the time they have really good tags or expression of GFP and things like that if you find the right one. So this is a really basic pcDNA 3.1. What we have here is a CMV promoter. That's a very strong mammalian promoter. There is also T7 promoter, so if you wanted to express your gene of interest in E. coli, for example, that promoter would do that. Here's our multiple cloning site with all of our restriction sites, and then all the other components. There's ampicillin resistance to the plasmid, things like that. And we'll get into a little more what these guys mean as we're going on. But this is basically, if you input a vector sequence into Vector NTI, this is the kind of map you can get. The other great thing about Vector NTI is that it actually has a library of Invitrogen plasmids that you can search through and pull them up and open them, and they'll all be annotated like this and everything. So that's another nice thing. For this, I work with the protein GPX4 since I had the sequence available and I'd already cloned it into something. We're doing that as our example. So we're going to put GPX4 into pcDNA 3.1. You could just pretend that this is any gene of interest that you like. So you could just say it's whatever you're interested in. Again, reminder, we're going from a plasmid and an insert to a cleaved plasmid insert to a nice ligated full plasmid with your gene of interest inserted. Okay, so the first thing that we need to do is identify our restriction sites within our vector that we want to go into. So here I've highlighted the multiple cloning site in Vector NTI, and you can see it here. Anywhere that there's this squiggly line with a word above it, that's a restriction site. So this is a BAM site, a HinD3 site, EcoR1 site, all these sites. And now you can go back to NEB and look at how these cuts look. So I tend to pick ones that are going to make sticky ends. And you can also look at which ones will work well together when you digest them. But typically, if you pick anything within the multi-cloning site, it'll put your gene of interest in the right place so that it's right behind that promoter. You don't want to pop your gene of interest here because it's not going to express well. Okay. So some really important things that you need to keep in mind when you're picking a restriction site is also not just is it in the right spot in the plasmid, but will it work with your insert? So number one, the sites have to be in the destination vector only once. Cool thing Vector NTI does is it makes all of the sites that occur only once red. So don't pick any black sites here. You're going to be cutting your vector all over the place, right? Because there's multiple sites. The other thing that you have to think about is making sure that your restriction enzymes aren't going to cut up your insert. So this insert has an EcoR1 site within it. And that's going to happen because DNA sequences are going to have these little 5 nucleotide sections in them. So if you have an EcoR1 site there and you cut with EcoR1, you're going to break your insert in half. And when you ultimately put it into your vector, you're going to end up with a truncated form of your gene of interest. No good. So I've highlighted here all of the good are the restriction sites in the multiple cloning site. Bad is EcoR1 because it's in our gene of interest. But BAMH1 and HindD3 here would be really good restriction sites to use. So if we were going to look at this, we'd have a BAM site and a Hindi site here. This is a particular instance where there was another vector with a BAM or Hindi site. And what you can do is cleave it out of that other vector and make this new one. So this is one option you can have, is digesting your vector out of your insert out of a parent vector and then using that insert to put in. Now, the likelihood that you're going to have a parent vector where your gene of interest has been put in at the BAM1, BAMH1, and HindD3 sites is almost impossible. If you have that kind of luck, you don't need to be here. You can go somewhere else because this is easy. All you have to do is cut it and pop it in. What we need to talk about more is making our insert by PCR amplification. Most of the time, you're going to have either your gene of interest in a vector or in DNA, and you're going to have to make it with PCR amplification. And some of the things that are really important here is, number one, you're going to want your primers to associate with your gene of interest, and typically that's about 18 to 21 bases. But you also need to add in your restriction sites. If you just amplify your insert without adding your restriction sites, you're not going to be able to make your sticky ends to pop it into your vector. So what you'll typically do is, again, look at NEB and come up with what these restriction sites look like, add them to either end of your primers, and then typically you have to add a couple of like 3 to 6 base pairs at the end, the 5' end of these primers, so that when you make your, when you ultimately make your PCR product and cleave it, you have higher cleavage efficiency. Restriction enzymes don't like to cleave right on their, right on the 5' end of their site. So you have to add a few more nucleotides, and that can be random. It doesn't really matter what you add there. You just want to make sure that they are not going to cause any hairpin structures in your final product. Okay, so in order to design this, this insert, you can use vector NTI, which is great. So if you put your gene of interest into vector NTI, and then you go to primer design, amplify selection, and then you've highlighted your gene of interest, so it's going to amplify that. You ask it to amplify the entire thing from 1 base pair to 820 base pairs. But you can add a 5' terminus to each of your primers. So for your sense primer, which would be amplifying this 5' end primarily first, you would add that BAMH1 restriction site. To your antisense primer, you're going to be adding the HindD3 to that restriction site to your primer. And ultimately what it's going to do is design primers for you, which is really nice. So you've got your sense primer and your antisense primer, and at that point you can take these oligonucleotide sequences, submit them to Invitrogen, and have them produce your primers. Now, once you have your insert that you've made in silico with the different restriction sites on each end, and then of course you have your pcDNA 3.1 with your different cloning sites that you know you're going to go into, you can do an in silico cloning experiment. So basically, you can say what's going to happen when I put my insert into my vector. Is it going to turn out okay? So what you can do is go to the cloning tab in Vector and TI, and then go to the clone to seek. So I put it bigger here. And you open up your vector, you open up your insert, and then all you have to do is follow the instructions at the bottom. So basically what I did is I selected from BAM-H1 to Hindi3, so that's all the vector that I want to keep, and then I selected from BAM-H1 to Hindi3 in my insert. So I'm going to be cloning from the BAM-H1 to Hindi3 site in both of these. Okay? Press clone, and what it makes is a nice new plasmid. So what I have here between my BAM-H1 site and my Hindi3 site is my GPX4 insert, and this is occurring after the CMV promoter and the T7 promoter, so it's where it needs to be. And it looks good. But we need to make sure that this is in frame, that there's not a frame shift mutation. So what can happen if you insert your gene of interest into a plasmid is that if there's an extra few nucleotides that are in the plasmid, and this can happen all the time, is that when you cleave at your site, you end up moving your start site a little, 1 or 2 nucleotides to the left or right, and you don't get your methionine at your start site. So what's best to do is highlight your entire plasmid in Vector and TI and say translate. And then you want to look right past that BAM-H1 site and make sure that your ATG is in frame and you're translating methionine. And then you can even look up your gene of interest, what the amino acid sequence is, and make sure that it's all how it should be. If you do happen to have an instance where you've got a frame shift, you can design your primers a little differently. So you can, if you're off 1 nucleotide, you can add 1 nucleotide between your cleavage site and your start site in your primer. And that will ultimately shift everything so you get rid of your frame shift. They also make plasmids in 3 different forms so that if you are seeing that there's a frame shift, you can pick a different one that's going to have 1 less nucleotide so that you don't get the frame shift. Okay, and then I'm just going to get back to this really quickly because it is an option and it's a very nice option. If you have those small inserts, this is, for example, the siRNA, so my the RNA sequence that could cause RNA interference. You've got a loop structure or a loop, a hairpin turn in there that has been added to the sequence, and then you can add your restriction sites to either side of that and have them produced by Invitrogen and make them, hybridize them, and then cut them. And ultimately what you're going to get is an insert that you didn't have to do PCR amplification of. And you can just purchase those oligos, and it's really simple. Okay, so we've planned it. We know that we've got our insert and it can go into our vector, and we know that there's no frame shift mutation. So all of this is just in silico. You haven't actually done anything towards your final goal. So now we have to go towards the final goal, so we have our handiwork ahead of us. So you've probably been wondering, well, she keeps saying plug a sequence into Invitrogen and you can get oligos. Okay, so this is the Invitrogen website, and you can order value oligos, and I really was serious about that. You put in an oligo name, and then you put in your sequence. And I think it ends up being like, it's $5 per oligo. And you just have them ship you the oligo that they've produced. So when you make your primers, you're going to have to do that. Okay. And then you also want to perform your PCR. So you want to pick your polymerase, and you need it to be high fidelity, because if you can imagine, if there are any mistakes when you are PCR amplifying your insert, which could be 300 bases, those are going to show up in your final product and you'll have a mutation, and that's problematic. So always pick a high fidelity polymerase. Tons of companies have these. I've got a bunch of the companies listed on that piece of paper, so you can look into those. New England Biolabs in particular has its own high fidelity system for cloning. So make sure you get a high fidelity polymerase, and then what you're going to want to do for your actual PCR amplification is use the melting temperature of the portion of the primer that's actually binding to your gene of interest. Remember, we added that restriction site and a couple of extra amino acids. That doesn't need to be incorporated into your math when you're actually designing your primers. So what you would add is just that 18 to 21 nucleotides that bind to your gene of interest and determine your melting temperature based on that. And this stuff is really easy. When you buy the polymerase, it gives you all the reagents and the instructions for how to do it. The only thing you have to determine on your own is this melting temperature. And I've got it on your sheet, but you can go to this melting temperature calculator to find out how to do that. All right, so now we've got a insert that you've PCR amplified. We've got your vector that you've purchased, and we want to actually do our digestion with our restriction enzymes. So go to NEB, find out how to best perform the double digestion between BAMH1 and HindE3. Separately, digest your plasmid and your insert so that they'll both have those overhangs, exactly how you need them. And then you can double check your digest based on the fragments you get. So in vectoring TI, you can do a restriction, in silico restriction digest, and it will tell you if you cut at these two sites, how big your fragments are going to be. So what you'd want to do when you run a gel of this digest is, if this was our vector digest, you would have this very large 5,000 nucleotide band, and that's your portion of your plasmid that you want to keep. And then you have this really small band, and that's from the part that was cut out that you do not want to keep. Same thing with the insert. Ignore that shouldn't be there, but you're going to have a certain size. So in this case, it's 826 nucleotides. What you'll want to do is run these on a normal DNA gel, and then based on the size that you're expecting to have, cut out your bands. So this ensures that we don't end up religating our 18 nucleotides that was in here. You want to make sure you separate your plasmid or your vector from the insert that you've cut out of it. And similarly here, but mainly this is so you can get a nice pure portion of your PCR product. So you just cut it out with a scalpel, this little area that's the right size. You use a UV platform. So you put your gel on the UV platform and you can see where exactly your bands are located. Cut it out, and then QIAGEN has a really cool kit called a gel extraction kit. You basically can take this chunk of gel, and at the end of the day you have a nice piece of your product. And you'll have to purify it before you can do anything else with it, but QIAGEN has tons of products that help with this kind of process. Okay, so now you have your digested vector and your digested insert, so you have to ligate them. In this case, this is where we had our HindE3 digest. So you have these sticky ends hanging out. This would be perhaps your vector, this would be your insert. All you have to do is add a DNA ligase and ATP, and you can get annealing and phosphodiester bond formation, and now you've got a complete circular piece of DNA, your plasmid. Okay, so in our case, our plasmid had ampicillin resistance. So this is an antibiotic resistance. So we're going to put our plasmid into bacteria, and we're going to have the bacteria manufacture our plasmid. So they just produce tons of plasmid. And all you have to do is grow them on a plate that has ampicillin on it. So anything that has our plasmid in it is going to survive on that plate. And you'll get these nice little colonies of bacteria that should contain your perfectly made vector. Okay, so the most important part of the entire thing is this ultimate validation step, because you want to make sure that what you made is what you think you made. So you'll take those bacteria, those colonies, you'll just pick one, let it grow up still in that antibiotic, and then use another QIAGEN kit, a plasmid maxi mini midi prep kit, and you will isolate that plasmid DNA from the E. coli. And then if you have your plasmid, and again you went into vector NTI with your whole plasmid and saw a bunch of different restriction sites that would give you certain size products, you can cut and you can see, well, if I cut in these places, am I getting the right size products? But ultimately I think it's most important to do your sequencing, because if you incorporated a mutation in your PCR and that wouldn't be your fault, you wouldn't know it. So definitely there's the University of Pittsburgh DNA sequencing core you can go to, and you can sequence your final product. And then you ultimately want to see, well, can I overexpress my gene of interest, and does it have whatever else I need associated with it? Does it have the marker I need? Does it have the tag I need? So ultimately transfecting your cells and seeing that overexpression is a really important part. Okay, so now once it's all been made and validated, you can use it for experiments, and ultimately the cells should express your gene of interest if they take up the plasmid. So I've told you the difficult portions of cloning. This is how it was done a while back, and before that it was even worse because there wasn't vector NTI. But there are some new tricks that are really, really helpful with cloning, and they just involve going to Invitrogen, for example, and just ordering a full topo cloning kit. Okay, so one of the shortcut mechanisms of cloning is called topo cloning, and basically what they do is they give you your destination vector, and it's already attached to topoisomerase 1, so it is already ready to accept an insert. It doesn't need to be cut, nothing like that. You would make, as before, your PCR product, but you don't need restriction sites on it anymore. Okay? All you need is that TAC amplify PCR product. And then you put it into their mixture, and ultimately 5 minutes at room temperature you have your ligation complete and everything's ready to go. So much simpler. I told you everything else just to tell you this, but it's important to understand why it's happening the way it is. But this is a really nice method. And we have a little time, so I'll just show you the basics of how topo works in this little video.
Topo cloning technology revolutionized cloning to deliver unparalleled simplicity, efficiency, and speed. No matter how difficult the samples or downstream application, 95% recovery of your desired clone is possible in just 5 minutes. But just how does this technology work? Let's start in the lab, where we'll find our research scientist Jeff setting up a topo cloning reaction. He combines his PCR amplified product with the water, salt solution, and topo vector, all included in the kit, and he then mixes gently. While Jeff incubates the reaction for 5 minutes at room temperature, let's take a closer look at what's happening in the reaction. The key to topo cloning technology is the enzyme topoisomerase 1, which functions both as a restriction enzyme and as a ligase. Its biological role is to cleave and rejoin DNA during replication. It binds to the phosphate group attached to the 3' thymidine and cleaves the DNA strand, enabling the DNA to unwind. The enzyme then religates the ends of the cleaved strand and releases itself from the DNA. To harness the religating activity of topoisomerase 1, our topo vectors are supplied linearized and have topoisomerase 1 covalently attached to the 3' phosphate group at each end. When the topoisomerase 1 activated vector comes into contact with Jeff's linear DNA fragment, the DNA fragment is ligated into the vector with high efficiency, and the topoisomerase 1 enzyme is released. In this experiment, Jeff has amplified his DNA of interest using a proofreading polymerase with 3' to 5' exonuclease activity, because the accuracy of his sequence is crucial to his application. These polymerases generate blunt-end PCR products, so he is using our zero-blunt topo PCR cloning kit. It is not necessary to purify the PCR product prior to topo cloning. As with any PCR cloning technology, the cleaner your PCR amplified DNA, the higher the likelihood that you'll clone the correct DNA insert. Some researchers use a Taq polymerase that generates PCR products with 3' A overhangs. In this case, a topo TA cloning kit must...
Maybe we can stop there. So you can learn a lot more about the different kits that Invitrogen has available by just going to their website. They've got all kinds of information, all of these videos and things like that. But I basically thought it'd be a nice little overview of how simple this process can be with a kit like this. And what you can do is go to the Invitrogen website and you can look up any of their particular topo vectors and purchase a vector. I mean, I bought one recently that had a V5 tag on the N-terminus. So you can basically just have, I put my gene of interest into that vector, and very easy process. But again, the planning's still important. You have to make sure you know which PCR polymerase you're going to be using so that you know which kit to buy, but super easy to just go on the Invitrogen site and figure that out. And another cloning strategy that's really simple is the gateway cloning procedure. Basically, what you can do is take your gene of interest through PCR or cDNA library, all that kind of thing, and you clone it into an entry clone using these ATT sites. So basically, they again hijacked a bacterial system. This is, or actually, I think it's viral. It's a lambda phage recombination system. But basically, you put it into these ATT sites, and once you have this entry clone, you're able to pop your insert out of this entry clone and into any other vector you might want to. So it's a really nice kind of Lego block system, where once you have your gene of interest, all you have to do is do a simple reaction, and that's kind of explained a little bit more here, either a BP or an LR reaction, and it basically is going to move your entry clone either into your destination vector, or it's going to move it back from your from a donor vector into the entry clone. So basically, you've got these different reactions you can perform to move your gene of interest into and out of different gateway systems. And the really great thing about this is that there are places that have already made gateway open reading frame entry vectors. So if you are interested in overexpressing tyrosine hydroxylase in a gateway system, all you have to do is go to Genocopia, I have the hardest time saying that, and look through their entry clone search, and you can find probably your gene of interest within an entry clone. And then all you have to do is buy ultimately the vectors that you want to gateway clone it into, and you're good to go. So here, if I want a human, say GPX4, easy enough. And these are all of the different entry clones that I can buy for GPX4 that use that gateway entry system. So it's a really, really nice thing. You pay $300, but you've got this vector with your gene of interest, you don't have to do any PCR or anything like that, and you can just pop it into any destination vector that you choose. So it's another really cool tool. So now that you know the theory and the tricks to molecular cloning, you can decide the cloning method that you'll use to create your own expression in vector. And please, don't pay someone else to do it, because you can do it. And you're going to have that control, and you aren't going to spend as much money in the end, and ultimately you'll know what you have. You'll be able to trust what you have. So that's my grand important theme for this whole thing, is being able to do this yourself makes life a whole lot easier in the long run. And I know from joining a lab that got their plasmids from other places, that having someone in there that can make whatever you want is really nice. One other thing I didn't mention in this presentation, but I think it's really important, is site-directed mutagenesis. So once you have your gene of interest in a vector, it's very easy to use a site-directed mutagenesis kit and actually create mutations or create truncations of your protein of interest. So if you're interested in changing your protein in a little way, also write that down and think about it. But site-directed mutagenesis means you don't have to do more cloning. You can just change sites or areas of your gene of interest. Okay, so I have great cloning manuals and protocols here. Ultimately, you saw an example, but when it gets to the nitty-gritty and you have a question, you want an answer at hand. So you can go to the Agene plasmid cloning by PCR, so they have a really nice little system that you can use, a handbook that you can use, and it goes through it step by step. I actually took some of my images from that book. Also, Molecular Cell Biology 4th edition has a really good cloning primer. And if you want to watch a YouTube video on plasmid cloning, it'll make it simple and approachable. And then there is a portion of New England Biolabs called NEB Cloner, and it's got all kinds of information, products, troubleshooting, because they use that gateway program as well. And so you can find vectors, you can find the best cloning method you want to use, and things like that. But again, grab one of these sheets before you leave, because I don't expect you to write all these websites down. I've got them all in here. So basics of cloning, choosing your system, and then also the website for the HSLS where you can get vector NTI. With that, I would love to take questions. I didn't mention at the beginning that I wanted you to interrupt me, but sometimes I fly. So if you have any questions, please let me know. All right, fantastic. Thank you.
Thank you very much.